Hi everyone, welcome. If you're here tonight, chances are you're looking for something quiet, something calm, something that will let your mind wander gently through an interesting topic and slowly, softly, carry you off to sleep. That's exactly what this is for. Tonight, we're going to talk about cells, the tiny, invisible building blocks of every living thing on Earth, including you. Right now, as you lie there, your body is made of trillions of cells, each one quietly doing its work, breathing, communicating, producing energy. All of it happening without you having to think about it at all. And that's one of the most peaceful things about biology, really. So much happens without our awareness. So much is simply taken care of, quietly, reliably. So let's begin. Find a comfortable position. Let your body settle into wherever you are. Let your breath slow down a little. You don't need to do anything special. You just need to rest and listen. We're going to move slowly tonight. There's no rush. We'll take our time with each idea. We'll revisit things gently. And if your mind drifts, that's perfectly fine. That's actually the point. Let's start at the very beginning. A cell is the smallest unit of life. That's the classic definition. And it's a good one. Simple and clear. Everything that is alive, every bacterium, every plant, every mushroom, every fish, every human being, is made of cells. Some living things are made of just one cell. Others, like you and me, are made of trillions. The human body, in fact, contains somewhere around 37 trillion cells. That number is so large it's almost meaningless when you first hear it. 37 trillion. If you tried to count to 37 trillion at one number per second, it would take you over 1 million years. And yet here you are, all of those cells working together, making you possible. The word cell actually comes from the Latin word cella, which means a small room or a chamber. It was given this name by a scientist named Robert Hooke, way back in 1665. He was looking at a thin slice of cork under a microscope, and what he saw reminded him of the tiny rooms in a monastery where monks slept. Small enclosed spaces, rooms within rooms, cells. Robert Hooke didn't fully understand what he was looking at, of course. He saw the walls of dead cork cells, but not living ones. It would take another few decades before scientists began to realize that these tiny chambers were the fundamental units of all living things. That realization, that all life is made of cells, is called the cell theory, and it's one of the most important ideas in all of biology. Cell theory has 3 basic parts. First, all living organisms are made of one or more cells. Second, the cell is the basic unit of life. Third, all cells come from pre-existing cells. That third part is particularly interesting. All cells come from pre-existing cells. Life, in a sense, is an unbroken chain. Every cell in your body descended from other cells, and those cells descended from other cells, all the way back and back and back, billions of years. We'll come back to some of that history later. For now, let's just sit with the idea of the cell itself. This tiny enclosed world, a small room full of activity, surrounded by a delicate membrane, existing in the vast interior of a body, or a leaf, or a drop of pond water. Each cell is its own world, its own chemistry, its own rhythm. Now, not all cells are the same. There are, broadly speaking, 2 major categories of cells in the living world. The first is called a prokaryotic cell. The second is called a eukaryotic cell. These are a bit of a mouthful, but let's go through them gently. Prokaryotic cells are the simpler of the 2. They're found in bacteria and in a group of microorganisms called archaea. Prokaryotic cells don't have a nucleus. Their genetic material, their DNA, just floats freely inside the cell in a region called the nucleoid. Prokaryotic cells are also generally smaller, much smaller. A typical bacterium might be about 1 to 10 micrometers across. A micrometer is 1/1,000,000th of a meter. So we're talking about something genuinely tiny, invisible to the naked eye. And yet, bacteria are extraordinarily successful. They live everywhere. In the soil, in the ocean, in the clouds, on your skin. Inside your gut, they were the first living things to appear on Earth billions of years ago. And in terms of sheer numbers and variety, they still dominate the living world. Eukaryotic cells are more complex. They have a nucleus, a central compartment, enclosed in its own membrane, where the DNA is stored. They also have other specialized compartments called organelles. Each organelle does a specific job. Eukaryotic cells are generally larger than prokaryotic cells. They're found in animals, plants, fungi, and a vast group of single-celled organisms called protists. Your cells are eukaryotic. Every cell in your body has a nucleus tucked inside it, holding a complete copy of your DNA. The word eukaryote comes from the Greek, meaning true nut or true kernel, referring to that nucleus, that true center that contained core of genetic information. And prokaryote means before the nucleus, referring to the more ancient, simpler design. We'll spend most of our time tonight talking about eukaryotic cells, since those are the ones that make up your body. But keep in mind that prokaryotic cells are fascinating in their own right, simple and ancient, and incredibly resilient. Let's start at the outside and work our way in. Every cell is surrounded by a cell membrane. This is also called the plasma membrane. It's the boundary between the inside of the cell and everything outside. The cell membrane is extraordinarily thin. We're talking about 10 nanometers thick. A nanometer is 1/1000th of a meter. The membrane is so thin that if you stacked 1,000 of them on top of each other, the resulting layer would still be thinner than a single sheet of tissue paper. And yet this membrane is absolutely essential. It controls what goes in and what goes out. It's the gatekeeper of the cell. The structure of the membrane is something called a phospholipid bilayer. That's a long phrase, but let's break it down gently. A phospholipid is a type of molecule. It has two parts. One end is attracted to water. It's called hydrophilic, which means water-loving. The other end is repelled by water. It's called hydrophobic, which means water-fearing. In water, these molecules arrange themselves in a very specific way. They line up so that their water-loving ends face outward, toward the water, and their water-fearing ends face inward away from the water. And when you have two layers of them arranged this way, back to back, you get a bilayer, a double sheet. The result is a flexible, fluid, self-sealing membrane. If you poke a small hole in it, it can often repair itself. If you stretch it, it can flex. It's not rigid like a wall. It's more like a very thin dynamic fabric. Embedded within this membrane are all sorts of proteins. Some of them act as channels, allowing specific molecules to pass through. Some of them act as receptors, receiving signals from outside the cell. Some of them act as pumps, actively moving substances in or out against the gradient. The membrane is, in a sense, alive with activity, even though it's only 10 nanometers thick. Let's just pause here for a moment. You can breathe slowly. Right now, every cell in your body has a membrane doing this quiet, constant work, deciding what enters, deciding what leaves, maintaining the delicate chemistry inside. It's been happening your whole life. It will continue through the night, while you sleep. Now, plant cells and fungal cells have something in addition to the membrane. They have a cell wall. The cell wall is a sturdy outer layer that sits outside the membrane. In plants, it's made primarily of a material called cellulose. Cellulose is a kind of carbohydrate, a chain of sugar molecules linked together. It's strong and rigid, which is why plants can stand upright, why a tree trunk is hard, why the leaves of a plant hold their shape. In fungi, the cell wall is made of a different material called chitin. Chitin is also found in the shells of insects and crustaceans. It's tough and protective. Bacteria also have cell walls, though their composition varies. Some have thick walls made of a substance called peptidoglycan. Others have thinner, more complex walls. The differences in their cell walls actually help scientists classify bacteria, and they also help determine which antibiotics will work against them. Many antibiotics work by targeting the bacterial cell wall. They interfere with the construction of that wall. causing the bacterium to become unable to maintain its structure, unable to survive. Your own cells don't have cell walls, which is why these antibiotics can target bacteria without harming you. A gentle distinction, a small, elegant difference. Moving inside the cell now. At the center of a eukaryotic cell, you'll find the nucleus. This is the command center, the library, the place where the cell's genetic instructions are stored. The nucleus is enclosed by a double membrane called the nuclear envelope. This envelope is studded with small openings called nuclear pores. These pores control the passage of molecules in and out of the nucleus. Large molecules like proteins cannot simply pass through. They need to be escorted by specialized carrier proteins. Inside the nucleus, the DNA is organized into structures called chromosomes. Human cells have 46 chromosomes, arranged in 23 pairs. If you were to take all the DNA out of a single human cell and stretch it out in a single line, it would be about 2 meters long. 2 meters, from a cell that is perhaps 10 micrometers across. That's an extraordinary amount of information packed into an extraordinarily small space. The DNA is not floating loose inside the nucleus. It's carefully wound around proteins called histones. These histones act like tiny spools, allowing the long strands of DNA to be coiled up tightly and organized neatly. Inside the nucleus, you'll also find a structure called the nucleolus. This is where the cell makes ribosomes, small structures that will eventually leave the nucleus and do the work of building proteins. We'll talk about ribosomes a bit later. The nucleolus appears as a darker, denser spot within the nucleus when you look at cells under a microscope. It's not enclosed by its own membrane. It's just a region of particularly intense activity. The nucleus communicates with the rest of the cell by sending out molecular messages. The language of these messages is RNA, a molecule related to DNA that carries instructions from the nucleus to the rest of the cell. We'll come back to all of this in much more detail. For now, just picture the nucleus as a quiet, organized center, a protected library, a place where the most important instructions are kept safe. Let's spend a little time with DNA. DNA stands for deoxyribonucleic acid. It's a molecule that carries genetic information, the instructions for building and running a living organism. The structure of DNA was described in 1953 by James Watson and Francis Crick, building on the X-ray crystallography work of Rosalind Franklin and Maurice Wilkins. Watson and Crick described it as a double helix, 2 long strands wound around each other like a twisted ladder. The sides of the ladder are made of alternating sugar and phosphate molecules. The rungs of the ladder are made of pairs of smaller molecules called bases. There are 4 bases in DNA. Their names are adenine, thymine, guanine, and cytosine. We usually refer to them by their first letters, T, G, and C. The bases pair up in a specific way. A always pairs with T. G always pairs with C. This is called complementary base pairing, and it's absolutely fundamental to how DNA works. The sequence of these bases, the order in which they appear along the strand, is the genetic code. It's like a language, written in an alphabet of 4 letters. T, G, C, arranged in different combinations. These 4 letters spell out instructions for making proteins, for regulating the cell, for determining the characteristics of an organism. The human genome, the complete set of genetic instructions for a human being, contains about 3 billion base pairs, 3 billion pairs of letters arranged in a precise sequence. If you were to read the human genome aloud at 1 letter per second, it would take you about 95 years. And all of that information, packed into the nucleus of every cell in your body. It's quite a thought to carry with you as you drift towards sleep. So how does the cell use this information? The process begins with transcription. This is the step where the DNA is read and a copy of part of the instructions is made in a slightly different molecule. RNA. RNA stands for ribonucleic acid. It's very similar to DNA, but with a few differences. First, it's usually single-stranded, not double-stranded. Second, it uses a slightly different sugar. Third, it uses the base uracil U instead of thymine T. During transcription, an enzyme called RNA polymerase binds to the DNA at a specific starting point called the promoter. It then travels along the DNA strand, reading the sequence of bases, and building a complementary RNA strand as it goes. This RNA molecule, a copy of the instructions for building a specific protein, is called messenger RNA or mRNA. It's a message carrying information from the DNA library out to the protein-building machinery of the cell. Before the mRNA can leave the nucleus, it goes through some processing. Non-coding sections called introns are removed. Coding sections called exons are spliced together. A protective cap is added to one end. A long tail of repeated nucleotides, called a poly-A tail, is added to the other end. All of this happens quietly, efficiently, within the nucleus. Then the finished mRNA moves out through the nuclear pores and into the main body of the cell, where it will find a ribosome and deliver its message. This is happening right now in the cells of your body, quietly, continuously, genes being read, messages being written, instructions being delivered, all while you rest. Now the mRNA has arrived at a ribosome. Ribosomes are small structures, among the smallest organelles in the cell. They can be found floating freely in the cytoplasm, or attached to a network of membranes called the endoplasmic reticulum. We'll talk about that in a moment. A ribosome has two parts called subunits. There's a large subunit and a small subunit. Together they form a structure that can read the mRNA and build a protein. The process of reading the mRNA and building a protein is called translation. It's called translation because the information is being translated from the language of RNA, a sequence of bases, into the language of proteins, a sequence of amino acids. Here's how it works. The mRNA threads through the ribosome. The ribosome reads the mRNA 3 bases at a time. Each set of 3 bases is called a codon. Each codon specifies a particular amino acid, one of the 20 amino acids that are the building blocks of proteins. A second type of RNA molecule, called transfer RNA or tRNA, acts as the translator. Each tRNA molecule carries a specific amino acid and has a region that can match up with the corresponding codon on the mRNA. As the ribosome reads each codon, the appropriate tRNA comes in carrying its amino acid. The ribosome links the amino acids together in a chain, one by one, slowly and steadily. When the ribosome reaches a special codon called a stop codon, it knows the protein is complete. The chain is released. This chain of amino acids, this newly made protein, then folds into a specific three-dimensional shape. The sequence of amino acids determines how it folds. The shape of the protein determines what it does. Proteins are the workhorses of the cell. They do almost everything. They provide structure. They catalyze chemical reactions, in which case they're called enzymes. They send signals. They defend against disease. They transport molecules. They regulate genes. The diversity of what proteins can do is staggering, and all of it flows from the sequence of bases in the DNA. Let's talk about the endoplasmic reticulum, or, as it's often abbreviated, the ER. The endoplasmic reticulum is an elaborate network of membranes that extends throughout the cell. It's a bit like a system of interconnected tubes and flattened sacs. And it comes in 2 varieties, rough and smooth. The rough endoplasmic reticulum gets its name from its appearance. It looks rough under an electron microscope because it's studded with ribosomes. These ribosomes are busy making proteins that will eventually be secreted from the cell or inserted into the cell membrane or sent to specific compartments within the cell. As these proteins are made, they are threaded directly into the interior of the rough ER, where they undergo further processing and folding. Quality control happens here. Proteins that are misfolded are tagged for destruction. The smooth endoplasmic reticulum doesn't have ribosomes on its surface, which is why it looks smooth. It has different jobs depending on the type of cell. In many cells, the smooth ER is involved in the synthesis of lipids, fats, and fatty molecules. In liver cells, it plays an important role in detoxifying drugs and other harmful substances. In muscle cells, the smooth ER stores calcium ions, which are released when the muscle needs to contract. The endoplasmic reticulum is also physically connected to the nuclear envelope. It's as though the library and the factory are linked by a continuous corridor. Proteins made in the rough ER can travel from there to the next processing station, called the Golgi apparatus. The Golgi apparatus, named after the Italian scientist Camillo Golgi, who first described it in 1898. It looks, under a microscope, like a stack of flattened pancakes, a series of curved membrane-bound sacs stacked together. Biologists sometimes describe it as looking like a stack of pita bread or a pile of deflated balloons. Whatever the comparison, it's a remarkable and busy organelle. The Golgi apparatus receives proteins from the endoplasmic reticulum. They arrive in small membrane-bound packets called vesicles. The vesicles fuse with one side of the Golgi stack, the receiving side called the cis face. Inside the Golgi, the proteins are modified. Sugar chains are added. Proteins are cut or trimmed. Labels are attached that tell the cell where the protein should go. Then the proteins move through the stack, from one layer to the next, being further modified as they go. Eventually they reach the other side, the shipping side, called the transface, and are packaged into vesicles again. These vesicles then travel to their destinations. Some head to the cell membrane, where they fuse and release their contents outside the cell. This process is called exocytosis. Some head to lysosomes. Some are delivered to other organelles. The Golgi apparatus is, in a sense, the cell's post office. Things come and get sorted and labeled, and are sent on their way. It's a quiet operation, orderly, continuous, like a postal sorting facility, working through the night. Every cell produces waste, worn-out proteins, damaged organelles, remnants of things that have been broken down. The cell has a system for dealing with this. It involves organelles called lysosomes. Lysosomes are small membrane-bound sacs filled with digestive enzymes. These enzymes can break down almost anything biological: proteins, lipids, carbohydrates, nucleic acids. The interior of a lysosome is acidic, around pH 4.5 to 5.0. That's significantly more acidic than the rest of the cell, which is close to neutral. The digestive enzymes inside work best under these acidic conditions. The acidic pH also acts as a safety mechanism. If a lysosome were to rupture, if it leaked its enzymes into the rest of the cell, those enzymes would find themselves in a near-neutral environment where they wouldn't work as well. It's a built-in failsafe. Lysosomes receive their cargo in several ways. Sometimes they receive material from inside the cell, old organelles or proteins that need to be recycled. This process is called autophagy, which literally means self-eating. It sounds harsh, but it's actually a very tidy and important process. Old damaged components are broken down, and their building blocks are recycled. Lysosomes also receive material from outside the cell. When a cell engulfs a bacterium or a piece of debris, a process called phagocytosis, the resulting package eventually fuses with lysosomes, and the contents are broken down. In a sense, the lysosome is the cell's stomach, the place where things are digested, but also a recycling center, because the broken down components, the amino acids, the sugars, the fatty acids, are released back into the cell to be reused. Nothing is wasted. And now we come to one of the most famous organelles of all, the mitochondria. You may have heard the phrase, the mitochondria is the powerhouse of the cell. It's one of the most widely repeated facts in all of biology education. And it's true. Mitochondria are the organelles that produce most of the cell's energy. The energy currency of the cell is a molecule called ADP, adenosine triphosphate. When you want to do anything biological, contract a muscle, build a protein, pump ions across a membrane, you need ADP. Mitochondria take fuel, primarily glucose and oxygen, and convert that fuel into ADP. The process is called cellular respiration, and it's a remarkably efficient and elegant process. Let's walk through it gently. The process of cellular respiration happens in several stages. The first stage is called glycolysis. This happens in the cytoplasm, the fluid interior of the cell, outside the mitochondria. In glycolysis, one molecule of glucose is split into two smaller molecules called pyruvate. A small amount of ADP is produced in this step. The pyruvate molecules then enter the mitochondria. They're converted into a molecule called acetyl-CoA, which enters a cycle of reactions called the citric acid cycle, or the Krebs cycle, named after the scientist Hans Krebs who worked it out in the 1930s. The Krebs cycle is a series of chemical reactions that go round and round, each time producing a small amount of ADP and, more importantly, releasing electrons that carry energy. These energetic electrons are passed along a series of proteins embedded in the inner membrane of the mitochondrion, in a process called the electron transport chain. As the electrons move through the chain, their energy is used to pump protons across the membrane. This creates a difference in concentration on the two sides, a kind of stored energy, like water behind a dam. Then, the protons flow back across the membrane through a remarkable molecular machine called ADP synthase. As they flow through, the machine spins, literally rotates, and uses the energy of that flow to build ADP molecules. ADP synthase is one of the most beautiful structures in biology. A tiny, spinning molecular turbine, using the flow of protons to generate the energy currency of life. The whole process, glycolysis, the Krebs cycle, the electron transport chain, can produce up to about 30 to 38 molecules of ADP from a single molecule of glucose. That might not sound like a lot, but the cell runs through enormous quantities of ADP. At rest, your body uses and regenerates its own body weight in ADP every single day. A busy muscle cell might regenerate its entire stock of ADP several hundred times per minute. It's a continuous, cycling process. ADP is made, ADP is used, and the process begins again. Mitochondria have another unusual feature. They have their own DNA. This is not typical for organelles. Most organelles don't have their own genetic material, but mitochondria do. And their DNA is circular, like bacterial DNA, not linear like the DNA in the nucleus. This observation, along with others, has led scientists to conclude that mitochondria were once free-living bacteria. Billions of years ago, an early cell engulfed a bacterium. But instead of digesting it, the bacterium survived inside the cell. Over time, a relationship developed. The bacterium became the mitochondrion. This theory is called the endosymbiotic theory, and it is now very well supported by evidence. The mitochondria in your cells are, in a very real sense, descendants of ancient bacteria. Ancient guests that never left. Ancient partners. There's something quietly wonderful about that, if you let yourself think about it for a moment. We've talked about mitochondria in animal cells. Plant cells have those too, but plant cells have something else, something that animal cells lack entirely. Chloroplasts. Chloroplasts are the organelles where photosynthesis happens. They're found in the cells of plants and algae. They're what makes plants green. Inside a chloroplast, there are stacks of flattened membrane sacs called thylakoids. These thylakoids are arranged in stacks called grana. And within the membranes of the thylakoids are the molecules responsible for capturing light. Photosynthesis is, in essence, the process of turning light energy into chemical energy. Carbon dioxide from the air and water from the soil are combined, using the energy of sunlight to produce glucose and oxygen. The glucose becomes food for the plant. The oxygen is released into the atmosphere. The oxygen you're breathing right now, as you lie there listening, was produced by photosynthesis. It was made in chloroplasts, in the cells of plants and algae. Photosynthesis happens in two stages. The first stage, called the light reactions, captures the energy of sunlight. This happens in the thylakoid membranes. The second stage, called the Calvin cycle, uses that captured energy to build glucose from carbon dioxide. This happens in the fluid interior of the chloroplast, called the stroma. Like mitochondria, chloroplasts also have their own DNA. Also circular, like bacteria, also thought to be descended from ancient free-living bacteria. In this case, a type of photosynthetic bacterium called a cyanobacterium. So the chloroplasts in a leaf cell are descended from ancient bacteria that learned to capture the sun. They became part of a cell, and eventually became an integral component of plants and algae. And because of that ancient partnership, the entire living world, including us, has been fed and oxygenated for hundreds of millions of years. The cell isn't just a bag of water with organelles floating around. It has structure, shape, internal architecture. This architecture is provided by the cytoskeleton. The cytoskeleton is a network of protein filaments that runs throughout the cell. It gives the cell its shape. It allows the cell to move. It helps organize the organelles. It plays a role in cell division. There are three main types of cytoskeletal filaments. The first are called microfilaments or actin filaments. They're the thinnest of the three, made of a protein called actin. They're important in cell movement and in changing the shape of the cell. In muscle cells, actin filaments interact with another protein called myosin to make the muscle contract. But we'll come back to that. The second type are called intermediate filaments. They're, as their name suggests, intermediate in size between the other two types. They're strong and stable, good for providing structural support. They're found in large numbers in cells that are under physical stress, like skin cells and cells that line the gut. The third type are called microtubules. These are the largest of the three. They're hollow tubes made of a protein called tubulin. Microtubules are involved in moving things around inside the cell. They form the tracks along which molecular motors carry cargo. They also form the spindle, the structure that pulls chromosomes apart during cell division. The cytoskeleton is dynamic. It's not static like the skeleton of a building. Filaments can be assembled and disassembled rapidly. The cell can reorganize its internal structure in response to signals, in response to changes in the environment, in response to the need to divide or move. It's constantly in motion on a molecular scale, even in a cell that appears to be sitting still. Inside the cell, there's a constant traffic of small membrane-bound packages. These are called vesicles. Vesicles are essentially tiny bubbles, small spheres enclosed by a membrane. They're used to transport molecules from one place to another within the cell. When the endoplasmic reticulum wants to send a protein to the Golgi apparatus, it packages it in a vesicle. The vesicle buds off from the ER membrane, travels through the cytoplasm, and fuses with the Golgi membrane, delivering its cargo. When the Golgi is finished processing a protein and wants to send it to the cell surface, it packages it in a vesicle. The vesicle travels to the cell membrane, fuses with it. The contents are released outside the cell. This process of releasing contents outside the cell by vesicle fusion is called exocytosis. The reverse, taking material in from outside the cell by forming a vesicle, is called endocytosis. Vesicles move along the cytoskeleton, particularly along microtubules. They're carried by molecular motor proteins called kinesins and dyneins. These motors literally walk along the microtubule tracks, one small step at a time, carrying their vesicle cargo. Kinesin steps along a microtubule the way you might walk down a long hallway, one foot in front of the other, step by step. But at the molecular scale, this happens millions of times per second. The whole system of membrane-bound compartments and vesicular transport in eukaryotic cells is called the endomembrane system. It includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, and the cell membrane, all connected by the continuous flow of vesicles, a quiet, orderly flow of packages, moving through the cell, delivering what is needed, picking up what needs to go. Cells don't live in isolation. They communicate, they send signals to each other, and receive signals from the outside world. This process is called cell signaling, and it's enormously complex and sophisticated. Let's walk through the basics gently. The most common type of signaling involves a molecule called a ligand. A ligand is a signaling molecule. It might be a hormone, a neurotransmitter, a growth factor, or any number of other molecules. It's produced somewhere in the body and travels, often through the bloodstream, to a target cell. When it arrives at the target cell, it doesn't usually enter the cell. Instead, it binds to a receptor protein on the cell's surface. The receptor sits in the cell membrane, waiting. When the ligand binds, it changes the shape of the receptor, and that shape change triggers a response inside the cell. This response is transmitted through a series of molecules called a signal transduction pathway. One molecule activates another, that one activates another, and so on, like a chain of falling dominoes, until the signal reaches wherever it needs to go. Sometimes the signal reaches the nucleus, where it causes certain genes to be turned on or off. Sometimes it triggers the cell to divide. Sometimes it triggers the cell to release a chemical. Sometimes it tells the cell to self-destruct, a controlled programmed cell death called apoptosis. Apoptosis. Let's pause on that for a moment. Programmed cell death sounds alarming, but it's actually essential. It's how your body sculpts itself during development. In a growing embryo, the cells between your fingers are removed by apoptosis. Without it, your hand would be a paddle. It's also how your immune system eliminates dangerous cells. Cells that have been infected by a virus. Cells that might become cancerous. They're triggered to die in a controlled, orderly way, without spilling their contents, without causing inflammation. Apoptosis involves a cascade of molecular events. Proteins called caspases are activated one after another, until eventually the cell's DNA is fragmented, its membranes are broken down, and the cell neatly packages itself into small fragments that are swallowed by nearby cells. It's a quiet ending. Every cell in your body began as a single cell, a fertilized egg, and from that single cell, through countless rounds of division, 37 trillion cells were eventually produced. Cell division is controlled by a series of events called the cell cycle. The cell cycle has two main phases. The first is called interphase. The second is called mitotic phase. Interphase is actually quite long. It's the phase where the cell grows, does its normal work, and prepares for division. It has three parts, G1, S phase, and G2. During G1, the first gap phase, the cell grows and carries out its normal functions. It monitors signals from its environment. Is there enough nutrition? Are conditions right for division? During S phase, the synthesis phase, the cell copies all of its DNA. Every single base pair. All 3 billion of them. Both copies are now kept in the nucleus, ready to be separated. During G2, the second gap phase, the cell continues to grow and prepares the machinery needed for division. All of this preparation happens quietly during interphase. The cell is busy, but it doesn't look particularly dramatic under a microscope. It just looks like a normal cell going about its business. Then comes the mitotic phase. Mitosis is the process of nuclear division. It's the stage where the duplicated chromosomes are carefully separated into two identical sets. Mitosis is traditionally divided into several stages: prophase, prometaphase, metaphase, anaphase, and telophase. These are a series of gradual, continuous changes that happen to the chromosomes and the cell. Let's move through them softly. In prophase, the chromosomes condense. The long, thin threads of chromatin coil up tightly, becoming short and thick. If you were watching a cell under a microscope, the chromosomes would become visible at this stage, dark, distinct shapes forming in the nucleus. At the same time, the mitotic spindle begins to form. those microtubules organizing into a structure that will eventually pull the chromosomes apart. In prometaphase, the nuclear envelope breaks down. The membrane that surrounded the nucleus dissolves away. The chromosomes are now free in the cytoplasm. The microtubules of the spindle reach in and attach to the chromosomes at a special region called the kinetochore. In metaphase, all the chromosomes line up in the middle of the cell. This middle line is called the metaphase plate. Each chromosome is being pulled from both sides. The spindle fibers from each pole of the cell tugging on opposite sides of each chromosome. A kind of tension. In anaphase, the tension is released. The chromosomes are pulled apart. The two copies of each chromosome, called sister chromatids, separate and move toward opposite poles of the cell. If you were watching this happen, it would look like the chromosomes are being dragged to the two ends of the cell, slowly, steadily. In telophase, the chromosomes arrive at the poles. A new nuclear envelope forms around each set. The chromosomes begin to relax and uncoil. Two new nuclei have formed. And then, following mitosis, the cell itself divides. This physical division of the cytoplasm is called cytokinesis. In animal cells, the cell membrane is pinched in the middle, like squeezing a balloon, until it splits into two separate cells. Two new cells, each with a complete, identical set of chromosomes, each ready to begin their own lives. The whole process of mitosis takes somewhere between 30 minutes and a few hours, depending on the type of cell. It's happening constantly in your body, in your skin cells, in your gut lining, in your bone marrow. A quiet, continuous renewal. The cell cycle isn't just allowed to run freely. It's carefully regulated. At key points in the cycle, there are checkpoints. These are moments where the cell checks to make sure everything is going correctly before continuing. The G1 checkpoint checks whether the cell is large enough, whether nutrients are available, and whether the DNA is undamaged. If something is wrong, the cell pauses. The G2 checkpoint checks whether DNA replication was completed correctly, and whether the cell is ready for division. The spindle checkpoint checks, during mitosis, whether all the chromosomes are properly attached to the spindle before anaphase begins. These checkpoints are controlled by proteins. There are two key types, cyclins and cyclin-dependent kinases, or CDKs. Together, they form complexes that activate each other and drive the cell forward through the cycle. There are also proteins that act as brakes, called tumor suppressors, and proteins that drive the cell to divide, called proto-oncogenes. In a healthy cell, these opposing forces are balanced. The cell divides when it should, pauses when it should, dies when it should. In cancer, this balance is disrupted. Mutations in key genes can disable the checkpoints. A mutated tumor suppressor gene no longer applies the brakes. A mutated proto-oncogene, now called an oncogene, drives the cell to divide continuously, ignoring the normal signals. The cell divides when it shouldn't. It doesn't die when it should. It accumulates more mutations, and eventually it can form a tumor, a mass of cells that have lost the normal controls on division. This is a process that typically takes many years and many mutations. It's not usually a sudden event. It's a slow drift away from the normal, and the body has many defenses. Checkpoints, DNA repair systems, immune surveillance. Most abnormal cells are caught and eliminated before they can become cancerous. We don't need to dwell on this for too long. The main point is that the normal cell cycle is tightly, beautifully regulated. A careful, deliberate process. And when it works correctly, as it does in the vast majority of cells, every single day, it is a remarkable example of molecular order and precision. Before we leave the topic of cell division, let's touch briefly on another type, meiosis. Meiosis is the type of cell division that produces sex cells, eggs and sperm. It's different from mitosis in several important ways. Whereas mitosis produces two identical cells with the full complement of chromosomes, meiosis produces 4 cells, each with half the number of chromosomes. Why half? because when an egg and a sperm join together at fertilization, they combine their chromosomes. If egg and sperm each had the full 46 chromosomes, the fertilized egg would end up with 92, and the next generation would have 184, and so on. The chromosome number would double with every generation. Meiosis prevents this by having the chromosome number. So egg has 23, sperm has 23. Together they make 46. Meiosis also involves something called crossing over. During meiosis, homologous chromosomes, the pairs of chromosomes you inherited from each parent, come together and exchange segments. They literally swap portions of their DNA. This shuffling of genetic material is one of the main sources of genetic variation. It's why siblings who have the same parents can be quite different from each other. Every egg and every sperm is genetically unique. This variation is one of the things that makes evolution possible. Without variation, there's nothing for natural selection to act on. Variation is the raw material of evolution, and meiosis, with its shuffling and having, is a major source of that variation. Your body contains many different types of cells. Not all cells in your body look or behave the same way. Even though they all contain the same DNA, they express different genes, and so they become different types. This process of cells becoming different types is called differentiation. Red blood cells, for example, are highly specialized. Their job is to carry oxygen from the lungs to the rest of the body. They contain enormous amounts of a protein called hemoglobin, which binds oxygen. In fact, red blood cells are so full of hemoglobin that, as they mature, they actually lose their nucleus. They give up their DNA and their ability to divide to make more room for hemoglobin. A mature red blood cell is essentially a small, flexible bag of hemoglobin. No nucleus, no mitochondria, just hemoglobin, and the machinery to keep functioning for about 120 days. After that, they're recycled in the spleen and liver. Nerve cells, neurons, are another extraordinary example of specialization. Neurons can be extraordinarily long. A single neuron that runs from your spinal cord to your foot can be over a meter in length. Most cells are measured in micrometers, but neurons can be measured in meters. Neurons transmit electrical signals. They communicate with each other through junctions called synapses, the electrochemical signals that allow you to think, feel, move, remember. All of those happen through the activity of neurons. Muscle cells are specialized to contract. They're filled with proteins, actin and myosin, that slide past each other when the muscle is activated, shortening the cell and generating force. A bicep muscle contains thousands of muscle cells working together, each one contracting slightly, the combined effect of their contractions lifting your arm. Liver cells are generalists in some ways. They do a huge variety of tasks: detoxifying the blood, making bile, regulating glucose levels, producing proteins for the blood. A liver cell is like a very busy factory worker. Skin cells, keratinocytes, produce large amounts of keratin, a tough structural protein. As skin cells mature, they move toward the surface of the skin, gradually filling with keratin, until eventually they die and become the hard, protective outer layer of skin. The outermost layer of your skin is, strictly speaking, made of dead cells. But new cells are constantly moving up from below, replacing the old ones. Every cell type is defined not just by what it does, but by which genes are active within it. The genome is like a complete recipe book. A red blood cell uses certain recipes. A neuron uses different recipes. A muscle cell uses others. But the book itself, the DNA, is the same in every cell. How do cells become specialized? The process begins very early in development. When the egg is fertilized and begins to divide, the early cells are capable of becoming any type of cell in the body. These are called totipotent cells, from the Latin for capable of all things. As development proceeds, cells begin to lose this total flexibility. They become committed to certain pathways. But there are still cells in the body that retain a remarkable degree of flexibility. Cells that can divide and give rise to many different cell types. These are called stem cells. Embryonic stem cells, from very early embryos, are pluripotent, capable of becoming almost any cell type in the body. Adult stem cells, found in specific tissues in the mature body, are more restricted, but still capable of producing a range of cell types within their tissue. Bone marrow, for example, contains hematopoietic stem cells that continuously produce all the different types of blood cells, red blood cells, white blood cells, platelets, throughout your life. In the brain, there are neural stem cells that continue to produce new neurons in certain regions, even in adulthood. This was once thought impossible. Scientists believed that adult brains couldn't produce new neurons, but it turns out they can. A small-scale, ongoing process of renewal. The study of stem cells has become enormously important in medicine. If we could understand how to guide stem cells to become specific cell types, we might be able to repair damaged tissues, replace lost neurons in Parkinson's disease, regenerate damaged heart tissue after a heart attack, restore insulin-producing cells in diabetes. The science is still developing, but the possibilities are significant. Cells in your body are not floating freely. They are surrounded by a scaffolding, a complex network of proteins and other molecules that gives tissues their structure, and that cells use to communicate with and anchor themselves to. This scaffolding is called the extracellular matrix. The extracellular matrix contains several key components. Collagen is one of the most important. It's the most abundant protein in the human body. Collagen fibers are strong and resist stretching. They give tissues their tensile strength. Your skin, your bones, your tendons all contain large amounts of collagen. Elastin is another important component. As its name suggests, it's elastic. It allows tissues to stretch and then spring back. Your lungs, your arteries, and your skin all contain elastin, which allows them to flex without tearing. The matrix also contains a variety of other proteins and complex molecules called proteoglycans, which help retain water and give tissues a gel-like quality. Cartilage, for example, is rich in proteoglycans, which is why it has that smooth, slightly spongy texture. Cells attach to the extracellular matrix through receptor proteins in their membranes called integrins. These integrins don't just provide physical attachment. They also transmit signals between the matrix and the cell's interior, influencing the cell's behavior. When cells receive signals from the extracellular matrix, they can respond by changing their shape, by moving, by dividing, or by differentiating into a different cell type. So the matrix is not just passive scaffolding. It's an active participant in the life of the cell. Let's return to the topic of energy and look a little more carefully at how cells extract energy from glucose. We touched on this earlier: glycolysis, the Krebs cycle, the electron transport chain. Let's spend a little more time with each step. Glycolysis. The word means splitting sugar. It happens in the cytoplasm, outside the mitochondria. It doesn't require oxygen, and it's very ancient. It's found in almost every living organism, from bacteria to humans. It's thought to be one of the oldest metabolic processes on Earth. In glycolysis, one molecule of glucose, a 6-carbon sugar, is converted through a series of 10 chemical reactions into 2 molecules of pyruvate, which has 3 carbons each. The process produces a net gain of 2 molecules of ADP and 2 molecules of a molecule called NADH. NADH is an electron carrier. It picks up energetic electrons from the reactions and carries them to the next stage. From glycolysis, the 2 pyruvate molecules enter the mitochondria. There, each pyruvate is converted into a molecule called acetyl-CoA. This conversion releases 1 carbon as carbon dioxide, which is why you breathe out CO2. The acetyl-CoA then enters the citric acid cycle. The citric acid cycle is a cycle of 8 chemical reactions. In each turn of the cycle, The 2 carbons from acetyl-CoA are released as carbon dioxide. Electrons are picked up by carrier molecules, more NADH, and also a molecule called FADH2. The cycle turns twice for every molecule of glucose, once for each pyruvate. At the end of the citric acid cycle, most of the energy from glucose has been transferred to NADH and FADH2. Only a small amount of ADP has been produced directly. But now comes the big step, the electron transport chain. The NADH and FADH2 molecules carry their electrons to the inner membrane of the mitochondrion. There, the electrons pass through a series of large protein complexes embedded in the membrane. As the electrons move through these complexes, their energy is used to pump protons, hydrogen ions, across the membrane from the interior of the mitochondrion to the space between the 2 membranes. This builds up a concentration difference, a reservoir of protons. Then the protons flow back through ADP synthase. This flow drives the synthesis of ADP. It's called the chemiosmotic mechanism. And the scientist who figured it out, Peter Mitchell, won the Nobel Prize for it in 1978. The final step of the electron transport chain involves the electrons being handed off to oxygen, which accepts them, along with protons, to form water. This is why you need oxygen to breathe. Without oxygen, the electron transport chain stops. Without the electron transport chain, very little ADP is made. Without ADP, the cell can't function. The whole process is exquisitely organized, molecularly precise, and happening, right now, in every mitochondrion of every cell in your body. What happens if there isn't enough oxygen? When oxygen is limited, cells can still make some ADP through glycolysis. But the pyruvate doesn't enter the mitochondria in the usual way. Instead, it's converted into other products. This is called anaerobic respiration, or fermentation. In your muscle cells, when you exercise intensely and oxygen can't be delivered fast enough, pyruvate is converted into lactate. This is sometimes called lactic acid fermentation. The lactate that accumulates is partly responsible for that burning sensation you feel in muscles during intense exercise. When you stop exercising and oxygen becomes available again, the lactate is converted back to pyruvate and processed normally through the Krebs cycle. In yeast cells, the situation is different. Yeast convert pyruvate into ethanol and carbon dioxide. This is the basis of fermentation in brewing and baking. When yeast ferment sugars in grape juice, they produce ethanol, wine. When they ferment sugars in grain mash, they produce ethanol, beer or spirits. When they ferment sugars in bread dough, they produce carbon dioxide, which makes the bread rise. Fermentation has been used by humans for thousands of years. We discovered it long before we understood any of the molecular biology. The yeast were doing their cellular respiration quietly and reliably, and we made use of the results. We've talked about proteins a good deal tonight. Let's slow down and look at them a little more carefully. Proteins are the most structurally and functionally diverse class of molecules in the cell. They carry out almost every function you can think of in biology. All proteins are made of amino acids. There are 20 different amino acids. Each has the same basic structure: a central carbon atom with an amino group on one side and an acid group on the other, and a variable side chain that differs from amino acid to amino acid. These amino acids are linked together in chains, polypeptide chains, through bonds called peptide bonds. A typical protein might consist of a few hundred to a few thousand amino acids. The sequence of amino acids in the chain, determined by the DNA sequence, is called the primary structure. When the chain folds up, it forms regular, repeating structures in certain regions. Spiraling regions called alpha helices, and flatter, zigzagging regions called beta sheets. These regular structures are held together by hydrogen bonds and are called the secondary structure. The chain then folds further into its overall three-dimensional shape, the tertiary structure. This folding is driven by interactions between the side chains of the amino acids, some attracted to each other, some repelled, some forming strong chemical bonds. In some proteins, multiple chains come together to form a complex. This is the quaternary structure. Hemoglobin, the oxygen-carrying protein in red blood cells, is made of 4 chains, 2 alpha chains and 2 beta chains that associate together. The final shape of a protein determines what it can do. Enzymes have active sites, precisely shaped pockets that can bind to specific molecules and catalyze reactions. Antibodies have specific binding sites that recognize foreign molecules. Receptor proteins have binding sites that fit specific signaling molecules. Shape is everything. When a protein misfolds, fails to achieve its correct 3-dimensional structure, it can't function, and misfolded proteins can be dangerous. Several serious diseases, including some forms of Alzheimer's disease and Parkinson's disease, are associated with the accumulation of misfolded proteins. The cell has quality control systems to detect misfolded proteins and either help them refold correctly or target them for destruction. These systems, including groups of helper proteins called chaperones, are constantly monitoring the protein population of the cell. Enzymes deserve their own section. An enzyme is a biological catalyst. It's a protein, occasionally RNA, that speeds up a chemical reaction without being consumed by it. Almost every chemical reaction in the cell is catalyzed by an enzyme. Without enzymes, most of these reactions would happen too slowly to sustain life. How does an enzyme work? An enzyme has an active site, a specific region of the protein that binds to the molecule or molecules it will act on. These molecules are called substrates. When the substrate binds to the active site, the enzyme and substrate come together in a complex. The enzyme then facilitates the chemical reaction, breaking a bond, forming a bond, transferring a group of atoms. The products are released, and the enzyme is free to bind another substrate molecule. A single enzyme molecule can catalyze thousands of reactions per second, and a cell can contain thousands of different enzymes, each performing its specific reaction. Enzymes are incredibly specific. Most enzymes catalyze just one reaction, or a small family of related reactions. This specificity comes from the precise shape of the active site. Enzyme activity can be regulated. Some enzymes can be turned on or off by molecules that bind to them at sites other than the active site, called allosteric sites. When an allosteric molecule binds, it changes the shape of the active site slightly, making the enzyme more or less active. This is how the cell fine-tunes its chemistry, not just turning reactions on or off, but modulating their rate in response to conditions. Temperature and pH also affect enzyme activity. Each enzyme has an optimal temperature and pH at which it works best. Body temperature, 37 degrees Celsius, is the optimal temperature for most human enzymes. If your body temperature rises significantly, enzymes start to malfunction. That's part of why high fevers are dangerous. Let's talk about water. Water is the medium of life. The cell's cytoplasm is mostly water. The reactions of the cell happen in water. Water is transported, regulated, and vital. Water moves across membranes by a process called osmosis. Osmosis is the movement of water from an area of lower solute concentration to an area of higher solute concentration across a semi-permeable membrane. In other words, water moves toward wherever things are more dissolved, toward higher concentrations of dissolved molecules. This has important consequences for cells. If a cell is placed in a solution that has a higher solute concentration than the cell's interior, water will flow out of the cell. The cell will shrink. This is called crenation in animal cells, or plasmolysis in plant cells. If a cell is placed in a solution that has a lower solute concentration than the cell's interior, water will flow into the cell. The cell will swell. In animal cells, if this goes too far, the cell can burst, lyse. Plant cells are protected from bursting by their cell wall. When water flows in and the cell swells, the cell wall pushes back, generating a pressure called turgor pressure. Turgor pressure is what makes plant leaves and stems firm and upright. When a plant wilts, when it's dry and drooping, it's because the cells have lost water and turgor pressure has dropped. The cell membrane is not impermeable to water, but water can also move through specific channel proteins called aquaporins. These are remarkably efficient water channels. A single aquaporin can allow about 1 billion water molecules per second to pass through. Aquaporins are found throughout the body. They're particularly important in the kidneys, where they allow the reabsorption of large amounts of water from the urine, preventing dehydration. Let's take a little detour and visit a very specific type of cell, the immune cell. Your immune system is made of many different types of specialized cells. Together, they patrol the body looking for threats, bacteria, viruses, fungi, parasites, cancer cells, and eliminating them. The white blood cells, or leukocytes, are the main cells of the immune system. There are many subtypes. Neutrophils are the most numerous white blood cells. They're fast-responding first responders. When a bacterial infection occurs, neutrophils are quickly recruited to the site. They engulf bacteria through phagocytosis and destroy them. They're relatively short-lived. They die after a few days. Macrophages are larger, longer-lived phagocytes. They clean up debris, engulf bacteria, and also send signals to recruit other immune cells. They're found in tissues throughout the body, patrolling quietly. Natural killer cells, a lovely, rather dramatic name, are a type of lymphocyte that kills cells infected by viruses or cells that have become cancerous. They do this by releasing cytotoxic molecules that trigger apoptosis in the target cell. B cells produce antibodies. When a B cell encounters a foreign molecule, called an antigen, it can differentiate into a plasma cell that churns out large quantities of antibodies. An antibody is a protein shaped like a Y with binding sites that recognize and bind specifically to the foreign antigen. Antibodies can neutralize pathogens, mark them for destruction, or activate other parts of the immune system. T cells have several roles. Helper T cells coordinate the immune response, releasing signaling molecules called cytokines that activate other immune cells. Cytotoxic T cells directly kill infected or cancerous cells like natural killer cells. The immune system also has memory. Once you've encountered a particular pathogen, your immune system creates memory cells, long-lived cells that remember the pathogen. If you encounter it again, the memory cells respond much faster and more effectively. This is the basis of vaccination. A vaccine introduces your immune system to a harmless version of a pathogen, or just its antigens, so that memory cells are created without you having to suffer the actual disease. It's a remarkable system, quietly patrolling, quietly remembering, protecting you through your life. Let's spend a little more time with the neuron. The neuron is one of the most extraordinary cells in the body. Its job is to transmit information, electrical and chemical signals, through the nervous system. A typical neuron has 3 main parts: the cell body, which contains the nucleus and most of the organelles; dendrites, branching extensions that receive signals from other neurons; and the axon, a long, thin projection that carries signals away from the cell body toward other neurons or muscles or glands. The axon can be very long, as we mentioned. Some axons in the human nervous system are more than 1 meter in length. They extend from the spinal cord all the way to the tips of the toes. Many axons are wrapped in a sheath of a fatty material called myelin. Myelin is produced by special cells: Schwann cells in the peripheral nervous system, oligodendrocytes in the brain and spinal cord. The myelin sheath acts as electrical insulation, and it dramatically speeds up signal transmission. Signals in a myelinated axon can travel at speeds of up to 120 meters per second. That's about 270 miles per hour. Unmyelinated axons transmit signals much more slowly, about 0.5 to 2 meters per second. How does the neuron transmit a signal? At rest, the inside of the neuron is negatively charged compared to the outside. This difference in charge is called the resting membrane potential. It's maintained by the constant work of ion pumps, particularly the sodium-potassium pump, which uses ADP to push sodium ions out and potassium ions in. When the neuron is activated, channels in the membrane open, allowing sodium ions to rush in. The inside of the membrane becomes temporarily positive. This is the action potential. The action potential propagates along the axon like a wave. One region of the membrane depolarizes, which triggers the next region to depolarize, and so on, all the way to the end of the axon. At the end of the axon, the terminal, the action potential triggers the release of neurotransmitters. These are chemical messengers that diffuse across the tiny gap between one neuron and the next, the synapse. The neurotransmitters bind to receptors on the receiving neuron, which may trigger or inhibit an action potential there. And so the signal continues. Different neurotransmitters have different effects. Glutamate is excitatory. It tends to stimulate neurons. GABA is inhibitory. It tends to suppress activity. Dopamine, serotonin, acetylcholine, norepinephrine. Each plays a different role in different circuits and systems. Your thoughts, your memories, your sensations, your emotions. All of them arise from patterns of activity in neurons. Networks of neurons firing in precise patterns across vast and intricate circuits. It's cellular activity, quiet, electrical, chemical, and it generates the entire inner experience of being you. We mentioned red blood cells earlier. Let's visit them more quietly now. The red blood cell, the erythrocyte, it's one of the most numerous cells in your body. You have about 25 trillion of them, and they're constantly being produced. About 2 million new ones every second in the red bone marrow. A mature red blood cell has a distinctive shape, biconcave, like a disc with a dimple in the middle on both sides. This shape maximizes the surface area for gas exchange. It also makes the cell flexible, able to squeeze through the tiny capillaries that are sometimes narrower than the cell itself. The cell is filled with hemoglobin, about 270 million hemoglobin molecules per red blood cell. Each hemoglobin can carry 4 oxygen molecules, so each red blood cell can carry over 1 billion oxygen molecules. As the red blood cell passes through the lungs, the hemoglobin picks up oxygen, which binds to iron atoms at the center of each hemoglobin molecule. The cell becomes bright red, oxyhemoglobin. As it travels to the tissues, the oxygen is released into the cells that need it, and carbon dioxide may be picked up. The cell becomes a darker red, deoxyhemoglobin. Then it travels back to the lungs. The carbon dioxide is released. Oxygen is picked up again, round and round, through a circuit of about 20 seconds each time. In a single day, a red blood cell may travel through the body several thousand times. After about 120 days, the cell becomes worn out. Its membrane becomes less flexible. It gets caught in the spleen, a filter-like organ, and broken down. The iron from the hemoglobin is recycled and reused. A quiet, orderly life cycle, lived entirely in service to the body's need for oxygen. The liver is one of the most metabolically active organs in the body, and its cells, hepatocytes, are among the busiest and most versatile cells you have. A liver cell does a remarkable number of different things. It processes glucose. When blood sugar is high, after a meal, liver cells take up glucose and store it as glycogen, a branch polymer of glucose. When blood sugar drops, the glycogen is broken down and glucose is released back into the blood. It processes fats. The liver synthesizes cholesterol and fatty acids and packages fats into particles called lipoproteins that carry them through the bloodstream. It detoxifies the blood. The liver is one of the main organs that breaks down drugs, alcohol, and other potentially harmful substances. Enzymes in the smooth ER of hepatocytes chemically modify these substances, often making them more water-soluble, so they can be excreted in the urine. It makes bile. Bile is a substance that helps digest fats. It's made in the liver, stored in the gallbladder, and released into the small intestine after a meal. It synthesizes blood proteins. Many of the proteins in your blood, including albumin, which maintains the fluid balance of the blood, and clotting factors, which help stop bleeding, are made in the liver. It stores vitamins and minerals. The liver stores fat-soluble vitamins A, D, E, and K, as well as iron and copper. And perhaps most impressively, the liver has a remarkable capacity for regeneration. If up to 70% of the liver is removed, by surgery, for example, the remaining cells can proliferate and restore the liver to its original size within a few weeks. Most organs can't do that. The liver is unusual in its regenerative capacity, and it's because the cells, the hepatocytes, retain the ability to re-enter the cell cycle and divide, even in the mature adult liver. Throughout tonight, we've touched on various ways cells communicate. Let's bring some of those threads together, gently. Cells communicate in several different ways. Some cells are in direct contact and can pass signals through physical connections. In many tissues, cells are linked by junctions called gap junctions, channels that connect the cytoplasm of one cell directly to the cytoplasm of a neighboring cell. Ions and small molecules can pass directly through these channels. This allows rapid, coordinated communication. In the heart, gap junctions allow the action potential to spread rapidly from one muscle cell to the next, ensuring the heart muscle contracts in a coordinated wave. That's the heartbeat. Other forms of communication involve signaling molecules. Endocrine signaling uses hormones, molecules released into the bloodstream by endocrine glands, which travel to distant targets throughout the body. Insulin, for example, is produced by cells in the pancreas and travels through the blood to tell muscle, liver, and fat cells to take up glucose. Paracrine signaling involves signals that act locally, released by one cell to affect neighboring cells. Growth factors often work this way. Autocrine signaling involves a cell releasing a signal that acts back on itself. This can be important in regulating the cell's own activity. And it plays a role in some cancers where cells stimulate their own growth. Synaptic signaling is the fastest form, the direct point-to-point signaling at a synapse that we discussed in the context of neurons. In all of these cases, the signal is received by a receptor, which triggers an intracellular cascade, which leads to a response. The response might be a change in gene expression, a change in metabolism, a change in cell behavior. The cell is always listening, always sensing its environment, adjusting. Cells age. Over time, cells accumulate damage, DNA mutations, oxidized proteins, dysfunctional mitochondria, shortening telomeres, and this cellular aging contributes to the aging of the organism as a whole. Let's talk about telomeres. At the ends of chromosomes, there are repetitive sequences of DNA called telomeres. They function as protective caps, preventing the chromosome ends from being recognized as damaged DNA and from sticking to other chromosomes. Every time a cell divides, the chromosomes are copied. But there's a problem. The machinery that copies DNA can't quite copy the very ends of the chromosomes. So with each division, the telomeres get a little shorter. After many rounds of division, the telomeres become critically short. This triggers the cell to stop dividing permanently, a state called cellular senescence. or the cell may trigger its own apoptosis. This limit on the number of times a cell can divide is called the Hayflick limit, after the scientist Leonard Hayflick, who described it in the 1960s. There is an enzyme called telomerase that can add telomere sequences back onto the ends of chromosomes, extending the cell's replicative lifespan. Telomerase is active in stem cells, in reproductive cells, and, unfortunately, in most cancer cells, which is one reason cancer cells can continue dividing indefinitely. Beyond telomere shortening, cells accumulate other kinds of damage over time. Reactive oxygen species, byproducts of normal cellular metabolism, including respiration, can damage DNA, proteins, and lipids. The cell has repair and antioxidant systems to deal with this, but they can't catch everything. Damaged proteins accumulate. Mitochondria become less efficient. Autophagy, that process of cleaning up old components, becomes less effective. The result, over decades, is a gradual decline in cellular function. Tissues and organs become less efficient. The immune system becomes less responsive. The ability to heal and regenerate decreases. This is the biology of aging. Not a single process, but an accumulation of many small changes. Gradual, quiet, happening in every cell over years and years. Let's step back, now very far back, and think about the origins of the cell. Life on Earth began somewhere around 3.5 to 4 billion years ago. Perhaps earlier, we're not entirely sure. The earliest life forms were simple, single-celled organisms. Prokaryotes, the forerunners of bacteria and archaea. The earliest cells would have had a very basic structure. A membrane, some simple genetic material. The minimal machinery needed to carry out the basic functions of life. Obtaining energy, replicating the genetic material, maintaining structure. How did the first cells arise? This is one of the most profound questions in all of science. We don't have a complete answer, but we have clues. The building blocks of life. Amino acids, nucleotides, lipids, sugars can form spontaneously under the right conditions. The famous Miller-Urey experiment in 1953 demonstrated that amino acids could be produced from simple inorganic molecules, water, ammonia, methane, hydrogen, under conditions thought to mimic the early Earth. More recently, experiments have shown that nucleotides can spontaneously form short chains of RNA under the right conditions. And RNA is remarkable because it can not only carry information like DNA, but also catalyze chemical reactions like proteins. This has led to the RNA world hypothesis, the idea that life began with RNA molecules that could both store information and catalyze reactions. Over time, DNA took over the role of stable information storage, and proteins took over most of the catalytic functions. But RNA remains at the heart of many fundamental processes: transcription, translation, regulation. From those early, simple cells, billions of years of evolution have produced the extraordinary diversity of life we see today. Bacteria, archaea, the ancestors of modern eukaryotes. The endosymbiotic event, that ancient engulfment that gave rise to mitochondria and later to chloroplasts, was a pivotal moment. It gave eukaryotic cells far more energy, enabling them to become larger and more complex. Multicellularity, the evolution of organisms made of many cooperating cells, followed later, arising independently, many times in different lineages. From single cells to multicellular organisms, from simple biochemistry to nervous systems, immune systems, reproductive systems, from bacteria to fish to mammals to you. All of it built on the foundation of the cell. Let's return gently to water. The cell is mostly water. By mass, a typical animal cell is about 70% water. Water is not just a passive solvent. It actively participates in many biochemical reactions. Hydrolysis reactions, reactions that break chemical bonds, involve water. Condensation reactions. Reactions that form chemical bonds, release water. Proteins fold the way they do, partly because of how their amino acids interact with water. Membranes form the way they do, because of how phospholipids interact with water. Water has unusual properties that make it particularly suited to being the medium of life. Its high specific heat capacity means it can absorb or release a lot of heat without changing temperature much, which helps organisms maintain stable internal temperatures. Its high heat of vaporization means it takes a lot of energy to evaporate water, which is why sweating cools you effectively. Water is also an excellent solvent. Its polar nature means it can dissolve a wide range of ions and polar molecules. The ionic bonds and hydrogen bonds that hold water molecules together also allow water to interact with and solvate charged and polar solutes. And water is most dense at 4 degrees Celsius. This means that ice floats, a crucial property for life on Earth. If ice were denser than liquid water, it would sink in lakes and oceans as they froze from the top down, eventually freezing solid and eliminating aquatic life. We don't often think about these properties. Water is so ordinary, so ubiquitous. We drink it, bathe in it, stand under rain. But inside every cell, it is at the center of life itself. We've talked about proteins and nucleic acids. Let's touch on the other 2 major classes of biological molecules: carbohydrates and lipids. Carbohydrates are sugars, and they're polymers. The simplest carbohydrates, monosaccharides, include glucose, fructose, and galactose. Glucose is the primary fuel of the cell. 2 monosaccharides joined together form a disaccharide. Sucrose, table sugar, is glucose and fructose. Lactose, milk sugar, is glucose and galactose. Many monosaccharides joined in long chains form polysaccharides. Starch is a polysaccharide used by plants to store glucose. Glycogen is a polysaccharide used by animals, including us, to store glucose in the liver and muscles. Cellulose is a polysaccharide used by plants for structural support. Carbohydrates also have structural roles in the cell. The outer surfaces of cells are covered in a coating of carbohydrates, attached to proteins, forming glycoproteins, and lipids, forming glycolipids. This carbohydrate coating, the glycocalyx, plays a role in cell recognition, cell adhesion, and protection. Blood types, for example, are determined by specific carbohydrate structures on the surface of red blood cells. Type A blood has one type of carbohydrate. Type B has another. Type AB has both. Type O has neither. Lipids are a diverse group of molecules, defined not by their structure, but by their property. They're hydrophobic. They don't mix well with water. The most important lipids in the cell are the phospholipids, which form the membrane. We talked about those earlier. Fats, triglycerides, are long-term energy storage molecules. A gram of fat contains more than twice the energy of a gram of carbohydrate. Fat is stored in adipose tissue, in fat cells called adipocytes. Cholesterol is a lipid that's incorporated into cell membranes, where it helps regulate membrane fluidity. At body temperature, cholesterol helps keep the membrane from being too fluid or too rigid. Cholesterol is also the precursor to steroid hormones, including testosterone, estrogen, and cortisol. So despite its reputation, cholesterol plays essential roles in the body. Some cells can move. Not all cells, many are fixed in place. But some cells are remarkably mobile. White blood cells, for example, can squeeze through the walls of blood vessels and migrate through tissue toward sites of infection. They're guided by chemical signals, a process called chemotaxis. They sense a gradient of chemical signals and move toward the source. How does a cell move? The movement involves the cytoskeleton, particularly actin filaments. At the leading edge of a moving cell, actin filaments polymerize, grow longer, pushing the cell membrane forward, forming a protrusion. At the back of the cell, other parts of the cytoskeleton contract, pulling the rear of the cell forward. It's a kind of coordinated, flowing movement. The cell extends at the front, attaches to the surface, pulls itself forward, and detaches at the back. Some single-celled organisms, and some cells in the body, move by means of flagella or cilia. Flagella are long, whip-like projections that propel a cell by beating back and forth. Sperm cells, for example, have a single flagellum that propels them toward the egg. Cilia are shorter and more numerous. They look like tiny hairs covering the cell surface. In your respiratory tract, cells lining the airways have cilia that beat in coordinated waves, sweeping mucus and trapped particles upward and out of the airways. The motion of cilia and flagella is driven by motor proteins, dyneins, that slide microtubules past each other, causing the flagellum or cilium to bend. A sperm's flagellum beats about 10 to 20 times per second, remarkably fast for something driven by molecular motors. Every cell exists in a context. A cell in the liver is surrounded by other liver cells, by blood vessels, by connective tissue. It receives hormones through the blood. It's part of a tissue, which is part of an organ, which is part of an organ system, which is part of an organism. Cells respond to their environment constantly. They adjust their gene expression. They change their metabolism. They signal to their neighbors. When conditions change, when nutrients become scarce, when temperature rises, when the cell is damaged, the cell responds. These responses are collectively called the stress response, or heat shock response, depending on the stressor. Stress response proteins, sometimes called heat shock proteins or chaperones, are made in larger quantities under stressful conditions. They help protect proteins from misfolding. They help stabilize damaged proteins. They're a kind of emergency support system. The relationship between a cell and its environment also includes its relationship with microorganisms. Your body is home to trillions of microorganisms, bacteria, archaea, fungi, and viruses, that live on and in you. Most of them are in your gut. Together, they're called the microbiome. The cells of your gut and immune system are in constant, active communication with these microbial residents. They've co-evolved with them over millions of years. Many of these microorganisms are beneficial. They help digest certain foods, produce vitamins, train the immune system, and protect against harmful pathogens. Your cells, and the microbial cells living within you, are, in a sense, partners, working together, shaping each other. Life is not made of isolated cells. It's made of communities. Let's think about time. Different cells have very different lifespans. Red blood cells live about 120 days. Neutrophils, those first responding immune cells, live only a few days. The cells that line your gut turn over every few days. The inner surface of your small intestine is essentially renewed every 3 to 5 days. Skin cells, the outermost layer, are shed and replaced over the course of about 2 to 4 weeks. Liver cells typically live about a year or more, though the liver can replace them if needed. Bone cells turn over slowly. Your entire skeleton is gradually replaced over about 10 years. Neurons in many parts of your brain, however, last your entire lifetime. The neurons you were born with, many of them are still with you. They've been firing and forming connections for as long as you've been alive. This creates an interesting situation. Your body is, in a sense, a constantly renewing collection of cells. Most of the cells in your body today weren't there 10 years ago. And yet, you are still you. The continuity of identity persists despite the turnover of the physical stuff. What persists is not the individual cells, but the patterns, the connections, the organization. A river is never made of the same water twice. And yet, it's still the same river. Your body is a little like that. A continuous pattern of organization maintained through constant renewal. We've talked about how cells have the same DNA, but behave differently. The mechanism behind this involves something called epigenetics. Epigenetics is the study of changes in gene expression that don't involve changes to the DNA sequence itself. The DNA is the same, but which parts are read, which genes are active, can differ from cell to cell, and can be influenced by experience and environment. There are several epigenetic mechanisms. One involves the histones, those proteins that DNA is wound around. Chemical groups can be attached to the histones, modifying how tightly the DNA is wound. If the DNA is wound very tightly, the gene is inaccessible. It can't be read. If it's loosened, the gene becomes accessible and can be expressed. Another mechanism involves the direct modification of the DNA itself. Specifically, the addition of a methyl group to cytosine bases in certain regions of the genome. This DNA methylation typically silences gene expression. These modifications can be passed from a cell to its daughter cells when it divides. This is how a liver cell remains a liver cell, not because it has different DNA, but because it has an epigenetic pattern that keeps liver genes active and other genes silenced. Epigenetic patterns can also be influenced by the environment: diet, stress, toxins, exercise, social experience. All of these can influence gene expression by altering epigenetic marks. There's even some evidence, though it's still debated, that some epigenetic changes can be passed from parent to offspring, not through changes in DNA sequence, but through epigenetic marks that survive the reprogramming that normally happens during early development. This is a fascinating and still evolving area of science. We've touched on cancer and cell aging. Let's think a little more broadly about cells and disease. Most diseases involve cells behaving abnormally in some way. In infectious disease, foreign cells, bacteria, or non-living parasites, viruses, interfere with the normal function of the body's cells. Viruses, for example, insert their genetic material into host cells and commandeer the cell's machinery to make copies of themselves. The cell becomes a virus factory. In autoimmune diseases, the immune cells of the body mistakenly attack the body's own cells. In type 1 diabetes, the immune system destroys the insulin-producing beta cells of the pancreas. In multiple sclerosis, it attacks the myelin sheath of neurons. In genetic diseases, a mutation in the DNA leads to a non-functional or absent protein. Sickle cell disease, for example, is caused by a single mutation in the hemoglobin gene. This mutation changes one amino acid in the hemoglobin protein, causing the hemoglobin to polymerize under low oxygen conditions, distorting the red blood cell into a sickle shape. These distorted cells can block blood vessels and are prematurely destroyed. Cystic fibrosis is caused by a mutation in a gene that encodes a chloride channel, a protein that moves chloride ions across epithelial cells. Without this channel, the cells can't properly regulate the movement of ions and water across the epithelium, leading to thick, sticky mucus in the lungs and other organs. In metabolic diseases, the enzymes of the cell's metabolic pathways don't function correctly. Phenylketonuria, for example, is caused by a missing enzyme that normally breaks down the amino acid phenylalanine. Without this enzyme, phenylalanine accumulates and, in excess, is toxic to the brain. All of these diseases have their roots in cellular biology. Understanding cells, how they work, what can go wrong, is fundamental to medicine. And much of the progress in modern medicine has come from understanding cellular and molecular biology. New treatments, new drugs, new approaches to diagnosis. All of it is built on a foundation of understanding what cells do and how they do it. We've covered a lot of ground tonight. Let's slow down now and just reflect for a moment. A cell is small beyond imagination. Most human cells are about 10 micrometers across. 10 millionths of a meter. If you placed 100 cells side by side, the row would be about 1 millimeter long. And yet, within that tiny space, there is extraordinary complexity. DNA stored and read, proteins built and folded, energy harvested, signals sent and received, waste cleared, structure maintained, division orchestrated. The biochemistry of a single cell involves thousands of different types of molecules, engaging in thousands of different reactions, all happening simultaneously, and in careful coordination. And you are made of 37 trillion of these cells, each one doing its work, quietly, without any instruction from your conscious mind. The cell is, in a sense, the unit of life, the smallest thing that can be said to truly be alive. And in studying the cell, we are studying life at its most fundamental. It's a humbling thing to think about. Right now, as you lie still and quiet, your cells are busy. Your heart cells are contracting rhythmically, pulling in calcium ions, shortening, relaxing, shortening again. Your neurons are firing gently in the patterns of rest. Your immune cells are patrolling. Your bone marrow is making red blood cells. All of it without effort, without awareness, just happening, continuously, reliably. Let's take one more slow, quiet tour of the cell, just to review everything we've seen tonight. Gently, without rushing. We begin at the outside, the cell membrane, that thin fluid double layer of phospholipids, studded with proteins, controlling what enters and what leaves, a dynamic boundary between the cell's inner world and the outside. Just inside the membrane, the cytoplasm, a slightly gel-like solution of water and dissolved molecules, organelles floating here and there, vesicles moving along cytoskeletal tracks, the cytoskeleton running through it all, microfilaments, intermediate filaments, microtubules, giving the cell shape, providing tracks, enabling movement. Near the center, the nucleus, enclosed in its double membrane, nuclear pores standing guard. Inside, the DNA wound around histones, organized into chromosomes. The nucleolus making ribosomes. Just outside the nucleus, the rough endoplasmic reticulum. Rough because of all the ribosomes studding its surface. Those ribosomes reading mRNA, building proteins, threading them into the ER. Further away, the smooth ER. Quieter, without ribosomes, making lipids, processing. The Golgi apparatus, that stack of flattened sacs, receiving vesicles from the ER, processing their contents, labeling and packaging, sending vesicles on their way. Lysosomes, small, dense, acidic, waiting to receive cargo, waiting to digest. The mitochondria, bean-shaped, with their own inner membranes, the electron transport chain embedded in those membranes, ADP synthase spinning, energy being harvested from glucose, and in a plant cell, the chloroplasts, green with their stacks of thylakoids, capturing light, building glucose from carbon dioxide. All of these components, working together, in a space 10 micrometers across, and outside the cell, in a tissue, the extracellular matrix, the scaffolding of collagen and elastin and proteoglycans, the neighboring cells connected by junctions, the blood vessel nearby, delivering oxygen and nutrients, carrying away carbon dioxide and waste. A complete world, contained, organized, living. We're coming toward the end of our journey tonight. We've traveled from the cell membrane to the nucleus, from DNA to proteins, from ribosomes to Golgi to lysosomes, from glycolysis to the electron transport chain, from mitosis to meiosis, from neurons to red blood cells to immune cells, from the origins of life to the nature of aging, all of it circling around this single central theme, the cell. The cell is where life happens, not just in a metaphorical sense, in a literal, biochemical, molecular sense. The reactions of life happen in cells. The instructions of life are stored in cells. The work of life is done by cells, and you are a collection of those cells, 37 trillion of them, organized into tissues and organs and systems working together in a coordination so precise and so reliable that most of the time, you don't even have to think about it. Life takes care of itself. At the cellular level, life is an ongoing, self-maintaining process. Cells take in energy. They use that energy to do work. They maintain their own structure. They repair themselves. They reproduce. And in doing so, they sustain you. They sustain every living organism on Earth. It's a very old story, billions of years old, and it's still going on, right now, in every cell of every organism alive. As you drift towards sleep tonight, carry this with you, if you like. The quiet hum of cellular activity, the slow turning of the Krebs cycle, the steady pumping of ion channels, the gentle transit of vesicles along cytoskeletal tracts, the faithful replication of DNA, all happening inside you, all happening without your needing to do anything at all. You can rest. The cells will take care of things. We talked about the cell membrane earlier, but let's return to it one more time, because it really is worth dwelling on. The cell membrane is, in many ways, the most fundamental structure in biology. It is the boundary that makes a cell a cell. Without a membrane to enclose and separate, there is no inside and outside. No cell, no life as we know it. Think about what the membrane has to do. It has to be a barrier, keeping the cell's contents in, and the outside world out. But it can't be completely impermeable, or the cell would be isolated from all nutrients, all signals, all communication. So it has to be selectively permeable, open to some things, closed to others. And it has to be able to adjust, changing which things it allows in or out, in response to the cell's needs. This is accomplished, in part, by the proteins embedded in the membrane. There are thousands of different membrane proteins. Each one has a specific job. Some are channels, hollow proteins that allow specific ions to pass through. Potassium channels, sodium channels, chloride channels, calcium channels, each one selective for a specific ion. Some are transporters, proteins that bind to a specific molecule on one side of the membrane and carry it to the other side. Sometimes this is passive, moving molecules from high concentration to low. Sometimes it's active, moving molecules against the gradient, using energy from ADP. Some membrane proteins are receptors. They sit on the outside surface of the membrane, extending outward into the extracellular space, waiting to bind to specific signaling molecules. When a signaling molecule docks with a receptor, the receptor changes shape. That shape change is transmitted through the membrane and initiates a cascade of events inside the cell. Some membrane proteins are enzymes. They catalyze reactions right at the membrane surface. Some are structural, anchoring the cell to the extracellular matrix or connecting cells to each other. The variety is astonishing, and all of these proteins are embedded in a membrane that is, as we said, about 10 nanometers thick. The fluid mosaic model describes the membrane, a fluid lipid bilayer with proteins scattered through it, some spanning the full thickness, some associated with just one side. The proteins can drift and move within the fluid lipid layer, slowly, but they're not fixed in place. Actually, the membrane is more complex than the original fluid mosaic model suggested. We now know that certain regions of the membrane are more organized than others. There are lipid rafts, regions enriched in certain lipids and proteins, that may play specific roles in signaling and membrane trafficking. The membrane is not uniform. It has texture, structure, dynamic organization, and it's also asymmetrical. The two leaflets of the bilayer, the inner one facing the cytoplasm, the outer one facing the outside, have different compositions. Different lipids predominate in each leaflet. Different sugars are attached to proteins only on the outer leaflet. This asymmetry is carefully maintained. There are enzymes called flippases, floppases, and scramblases that control the movement of lipids between the two leaflets. One of the signals of early apoptosis, programmed cell death, is actually a disruption of this asymmetry. A specific lipid normally kept on the inner leaflet appears on the outer surface, where it signals to immune cells. This cell is dying. Come and clean it up. Even the arrangement of molecules in the membrane carries information. There are a few organelles we haven't visited yet. Let's stop in on them briefly. The peroxisome. Peroxisomes are small membrane-bound organelles, found in eukaryotic cells. They're involved in oxidative reactions that use oxygen. They break down fatty acids in a process called beta oxidation. They also detoxify certain harmful molecules, including hydrogen peroxide, which is itself a toxic byproduct of their oxidative reactions. To deal with hydrogen peroxide, peroxisomes contain an enzyme called catalase. Catalase breaks hydrogen peroxide down into water and oxygen very rapidly. In fact, catalase is one of the fastest enzymes known. It can process millions of molecules of hydrogen peroxide per second. In liver cells, peroxisomes play a particularly important role in breaking down alcohol. The vacuole. Vacuoles are large fluid-filled compartments, bounded by a membrane. They're found in both animal and plant cells, but are particularly prominent in plant cells. In plant cells, a central vacuole can occupy 80 to 90% of the cell's volume. It acts as a storage compartment for water, nutrients, and waste products. It also contributes to turgor pressure, the internal pressure that keeps plant cells firm. Animal cells have smaller vacuoles that serve various purposes: storage, transport, removal of waste. The centrosome. The centrosome is a small structure near the nucleus. It's the main microtubule organizing center of the cell. From the centrosome, microtubules radiate outward through the cell. During cell division, the centrosome duplicates. The two copies move to opposite poles of the cell and organize the mitotic spindle, the structure that separates the chromosomes. Each centrosome contains two structures called centrioles. These are short cylinders made of microtubules arranged in a specific pattern. The exact role of centrioles is still being studied, but they're found at the base of cilia and flagella, where they form structures called basal bodies. Let's visit the heart cell. The cells of the heart muscle are called cardiomyocytes. They're unlike other muscle cells in some important ways. Skeletal muscle cells, the ones that move your limbs, are under voluntary control. You think about lifting your arm, and neurons fire, and the muscle cells contract. Cardiac muscle cells in the heart don't work that way. They contract rhythmically on their own, without waiting for instructions. They're intrinsically rhythmic. A cardiomyocyte has a built-in tendency to generate electrical signals. Certain cells in the heart, particularly in a region called the sinoatrial node, or SA node, are the natural pacemakers. They spontaneously generate action potentials at a steady rate. Those action potentials spread through the heart, triggering the coordinated contraction of the heart muscle. The rate at which the SA node fires is modulated by the nervous system. The sympathetic nervous system speeds it up during exercise or stress. The parasympathetic nervous system slows it down during rest. But the heart can beat even without input from the nervous system. A heart removed from the body will continue to beat for a time, if given appropriate conditions. The pacemaker cells keep going. Cardiomyocytes are also unusual in that they're highly connected to each other. They're linked by gap junctions, allowing the action potential to spread rapidly and synchronously through the heart muscle. Every cell in the heart contracts at essentially the same time, ensuring the heart beats as a coordinated whole rather than a chaotic twitching. And those cardiomyocytes are working continuously, without rest, every second, every minute, every year of your life. If you live to 80 years of age, your heart will have beaten about 3 billion times. 3 billion contractions. 3 billion cycles of calcium entering the cardiomyocyte, activating the contractile proteins, shortening the cell, then being pumped back out. 3 billion times. Quietly, continuously. Right now, as you listen, let's make a brief stop at the kidney. The kidneys filter your blood. About 180 liters of fluid per day pass through the kidneys. From that, they produce about 1.5 liters of urine. The cells of the kidney are extraordinarily active. They are among the most metabolically demanding cells in the body. The kidney is organized into millions of tiny filtration units called nephrons. Each nephron has several distinct regions: the glomerulus, the proximal tubule, the loop of Henle, the distal tubule, and the collecting duct. The cells in each region have specific jobs. Cells in the proximal tubule reabsorb about 65% of the filtered water, along with glucose, amino acids, and ions. They do this by actively transporting solutes from the tubule back into the surrounding blood vessels, and water follows by osmosis. The cells of the loop of Henle create a concentration gradient in the kidney tissue, more concentrated deep in the kidney, less concentrated near the surface. This gradient allows water to be drawn out of the collecting duct, and thus the urine to be concentrated. The hormone antidiuretic hormone, ADH, regulates how permeable the collecting duct cells are to water. When you're dehydrated, more ADH is released, more aquaporins are inserted into the collecting duct cells, more water is reabsorbed, and your urine becomes more concentrated. When you drink a lot of water, ADH levels drop, fewer aquaporins are inserted, less water is reabsorbed, and your urine becomes more dilute. This is the cellular basis of kidney function: active pumping, selective permeability, hormonal regulation, all at the cellular level. In the back of your eye, in the retina, there are cells that can detect light. These are the photoreceptor cells. There are two types, rods and cones. Rods are sensitive to low light. They allow you to see in dim conditions, in twilight, in shadow. There are about 120 million rod cells in each human retina. Cones are sensitive to color. There are three types, each responding to different wavelengths of light, roughly corresponding to red, green, and blue. There are about 6 million cone cells in each retina, concentrated in the central region called the fovea. How does a photoreceptor cell detect light? The outer segment of a photoreceptor cell is packed with stacks of membrane discs. These discs are embedded with molecules called opsins, proteins that are bound to a light-sensitive molecule called retinal. When a photon of light strikes a retinal molecule, the retinal changes shape. This shape change activates the opsin. The opsin then triggers a cascade of molecular events, a signal transduction pathway, that ultimately changes the electrical state of the cell. The cell hypopolarizes, becomes more negative inside. This changes the amount of neurotransmitter it releases onto the next cell in the visual pathway. The signal is then processed through several layers of cells in the retina before being sent via the optic nerve to the brain. The brain then interprets these patterns of signals as visual images. All of vision, every scene you've ever seen, every face, every sunset, every page of text, began with light striking retinal molecules in the outer segments of your photoreceptor cells. The retinal molecule, after being activated by light, has to be regenerated before the cell can respond to another photon. This takes a moment, and it's why, after seeing a bright flash of light, you see an afterimage for a few seconds. The photoreceptors that were exposed to the brightest parts of the scene take longer to recover. We've talked about DNA as the repository of the cell's genetic instructions, but DNA is constantly under threat. UV radiation from sunlight can damage DNA. Certain chemicals can damage DNA. Even normal metabolic processes, the movement of electrons during respiration, can generate reactive molecules that attack DNA. It's estimated that each cell in your body experiences thousands of incidents of DNA damage per day. Thousands. And yet, most of this damage is repaired before it can cause a mutation or interfere with cell function. The cell has an elaborate toolkit of DNA repair systems. The most common type of DNA damage, caused by UV light, involves the formation of bonds between adjacent thymine bases. These thymine dimers distort the double helix and interfere with replication. The cell repairs these through nucleotide excision repair. An enzyme recognizes the distortion in the helix. A section of the DNA strand containing the damage, about 25 to 30 bases, is cut out and removed. The resulting gap is filled in by a DNA polymerase, using the opposite strand as a template. Then a joining enzyme called a ligase seals the final nick. Other types of repair include base excision repair, mismatch repair, and double-strand break repair. Each targets a different type of damage. Each involves a distinct set of molecular machinery. Double-strand breaks, where both strands of the double helix are broken, are particularly dangerous. If not repaired correctly, they can lead to chromosomal rearrangements. The cell has specialized repair pathways for these, including one called homologous recombination, which uses the sister chromatid as a template to repair the break accurately. Mutations in DNA repair genes are associated with increased cancer risk. People with mutations in certain repair genes are more prone to developing specific types of cancer, because their cells are less able to correct DNA damage before it becomes heritable. This underscores how central DNA repair is to cellular and organismal health. We've been on quite a journey. We began with the simplest idea. The cell is the basic unit of life. And from there, we moved through the membrane, the nucleus, the organelles, the proteins, the energy, the signaling, the division, the specialization, the communication. We visited neurons and red blood cells, liver cells and heart cells, immune cells and photoreceptors. We touched on evolution and aging, on disease and repair, on epigenetics and stem cells. And through all of it, we've seen one consistent theme. The cell is organized. It is purposeful. It has systems within systems, each one contributing to the whole. It is, in its quiet way, a kind of miracle, not in a supernatural sense, but in the sense that it is genuinely extraordinary, that something as small and as fragile as a cell can be so complex, so capable, so alive. And the miracle is multiplied 37 trillion times in your body alone. If you were to translate the activities of a cell into human terms, it would sound something like this. The DNA is the library. The books contain instructions for making everything the cell needs. The books are not removed from the library. They stay safe inside. But copies are made, mRNA, and the copies are sent out. The ribosomes are the factories that read the copies and build the products. The endoplasmic reticulum is the workshop where some products are assembled and prepared. The GOLGI is the shipping department, where products are packaged and labeled for delivery. The mitochondria are the power plants, converting fuel into electricity, or in this case, fuel into ADP. The lysosomes are the recycling centers, breaking down old materials and returning the components to be reused. The cell membrane is the wall of the building, with gates and receiving docks and security systems. The cytoskeleton is the scaffolding, the roads inside the building along which workers and cargo move. The nucleus is the boardroom, where the master plans are kept. The boardroom is surrounded by security, nuclear pores that control what comes in and what goes out. And the cell cycle is the schedule, the regular rhythm of growth, preparation, and division. And the checkpoints are the quality control inspections, making sure everything is in order before the next stage begins. All of this in a space 10 micrometers across, a building more complex than anything humans have ever constructed, in a space much smaller than you could see without a microscope. It's getting late. If you've been listening for a while, your breathing has probably slowed. Your thoughts have probably softened. The ideas we've been exploring, membranes and ribosomes and ADP and chromosomes, have probably blurred and blended and become more like a quiet background hum than a series of distinct facts. That's fine. That's exactly fine. The science will be there tomorrow. The cells will still be working. The DNA will still be being read. The ADP will still be cycling. None of it requires your attention to keep going. There's something genuinely comforting about that. In a world where so much seems to require constant attention and effort, the biology of your cells runs on without you. Tirelessly, reliably. Your heart cells don't need you to remind them to contract. Your immune cells don't wait for instructions before they go to work. Your neurons are already in the slow rhythms of sleep. If you're drifting off, slower oscillations, gentler patterns. Sleep is actually a time of important cellular activity in the brain. During sleep, the brain's waste clearance system, sometimes called the glymphatic system, becomes more active. Cerebrospinal fluid flows more freely through the spaces between brain cells, washing away metabolic waste products that accumulated during waking hours. Some researchers believe this is one of the main functions of sleep, cellular housekeeping in the brain, clearing out the day's debris, preparing for another day of activity. So even as you sleep, the cells are working, cleaning, repairing, consolidating, processing. Sleep is not a shutdown. It's a different mode of cellular activity, a quieter, more inward mode, but essential. Life has been on Earth for somewhere between 3.5 and 4 billion years. Think about that number. For billion years, the cells in your body are connected through an unbroken chain of cell divisions to those first cells. For billion years of cellular life, each generation passing to the next, all the way to you. The basic chemistry of the cell, ADP, DNA, ribosomes, membranes, is essentially the same across all life on Earth, from bacteria to humans. This tells us that all life on Earth shares a common ancestor, a last universal common ancestor, sometimes called LUCA. From that one ancestral cell, the entire diversity of life has arisen, all through the same basic process. Cells dividing, accumulating slight differences over time, adapting to different environments, eventually becoming distinct species. The bacteria in your gut are your very distant cousins. So are the cells of a mushroom, and an oak tree, and a whale, and a tardigrade, those remarkable microscopic animals that can survive freezing radiation and the vacuum of space. All of us, all life, are variations on a theme. Cells doing what cells do, with modifications, specializations, elaborations, but fundamentally the same. There is something very peaceful in that thought, if you let yourself sit with it. We are all cells. All of life is cells, all connected. The night is quiet now. Let's count down through the parts of the cell one more time, very gently, like counting backward from 10. The cell membrane, that thin, beautiful fluid boundary, keeping inside and outside. The cytoplasm, that gel-like interior, where so much happens. The endoplasmic reticulum, that network of membranes, rough and smooth, busy and quiet. The Golgi apparatus, those stacked curved sacks, sorting and shipping, doing their steady work. The mitochondria, those ancient guests, making ADP, spinning their tiny turbines. The lysosomes, small and acidic, doing the quiet work of digestion and recycling. The nucleus, at the center, holding the chromosomes, holding the DNA, holding the instructions. And outside the nucleus, inside the cell, everywhere at once, the ribosomes, reading, building, making proteins, making everything. Let all of these images dissolve now. Let them soften. Let them blur. You don't need to hold them. They'll be there tomorrow. For now, just breathe. In and out slowly. Your cells are breathing with you, taking in oxygen, releasing carbon dioxide, running their quiet chemistry. Everything is taken care of. The cells are working. You can sleep. Good night.